Hi, my name is Caeley Bryan, and I'll be presenting a paper entitled Melatonin Attenuates LPS-Induced Depressive-like Behaviors and Microglial NLRP3 Inflammasome Activation through the SIRT1 NRF2 pathway. This is by Rose et al., published in 2019. So to begin with a little bit of research background, so what is depressive-like behavior and what are the pathways that we use to create it? So there are four main ways that scientists create depression in animal models. They are acute or chronic stress exposure, gene-environmental interactions, exogenous administration of glucocorticoids, and genetic manipulation. The model that this paper is looking at is an inflammation or stress-induced depression model that is caused by LPS injection. Depressive-like behavior is often measured in behavioral assays that look at lack of locomotor activity and survival instinct. And it's important to understand depression in animal models because MDD, or major depressive disorder, affects approximately 350 million people worldwide, according to the World Health Organization. So to get a little bit more specific, we're going to look at that like microglial NLRP3 inflammasome. So what is that and why should you care? It's part of that innate immune response. It's activated by PAMPs or DAMPs, so that's pathogen-associated molecular patterns and danger-associated molecular patterns. So that's things like that lipopolysaccharide we just talked about, or something like extracellular ATP that shouldn't be there in your body. It releases inflammatory cytokines, more specifically IL-1 beta and IL-18. This will come up a lot later on. And it can also be triggered by reactive oxygen species. So let's go on to melatonin. It doesn't just help you sleep. It's a hormone from L-tryptophan that's released from your pineal gland, but it's also released in a lot of your peripheral tissues. It helps regulate circadian rhythms through modulating expression of clock genes. I'm sure that's what all of you know it for. But it also has been proved to be neuroprotective, anti-inflammatory, an antioxidant, and anti-apoptotic, which means it's kind of the perfect model for this sort of experiment. So then finally, let's talk about all these random letters there. It's the SIRT1 NRF2 pathway. So what is it? Why is it activated? So NRF2 is nuclear factor erythroid 2. We're just going to call it NRF2 for the rest of the presentation. It's a transcription factor that codes for genes that are antioxidant and anti-inflammatory. And then SIRT1 stands for silent information regulator 2, homolog 1. Again, we're just going to call it SIRT1. And its dysregulation has been shown in a lot of different major depressive disorder studies. And the pathway between those two together has been often shown to be neuroprotective, which is why it's kind of a target in an experiment like this. So the hypothesis for this experiment is that melatonin will reduce depressive-like behavior in LPS-injected mice through a reduction of NLRP3 inflammasome activity. So how did they do that? So they picked some female BALBC mice who were aged from 12 to 14 weeks. If you don't know what BALBC mice are, they're just an albino inbred strain of mice. They look a lot like the mouse to the right. And they had an N of 5 per group with 3 groups, a control group, an LPS group, and then an LPS and melatonin group. They evaluated depressive-like behavior through 2 different tests, a forced swim test and a tail suspension test, both of which are locomotor tests. And then they injected mice with LPS at 5 milligrams per kilogram IP or intraperitoneally. And they injected with melatonin at 30 milligrams per kilogram every 6 hours, starting 2 hours pre-LPS up to 24 hours post-LPS, at which point they sacrificed the mice after behavioral experiments. So if I went through that a little quick, here's a closer look. A is that timeline of injection. You can see that pre-treatment versus post-treatment. That becomes pretty important later on. And then in B and C, you can just see with that FST, decreased mobility with LPS injection that gets recovered with melatonin. Same thing in that tail suspension test as well. So moving on from those phenotypic results to look at some biochemical results. All biochemical experiments were done with the hippocampus. They used Western blotting to look for NLRP3 markers and caspase protein levels, and mRNA for NLRP3 was evaluated with ELISA kits. They also used immunofluorescence staining to look at microglial astrocyte and neurofilament markers. I'm really only going to talk about the microglial staining from now on, but you can see some of this astrocyte staining over here to the right in this first row, this first column more like here in figure A. That GFAP is an astrocyte marker, and you can see that there is heavy activation in the LPS row, but that isn't really shown in that melatonin and LPS row. Same with that NLRP3. So the toxicity of melatonin on microglia was determined with a lactate dehydrogenase activity assay and CCK8 cells. I'm not going to show a figure for this. It's not incredibly important, but doses below 500 micrograms were entirely non-toxic, and above that could cause cell death. So moving on from that, they used 500 micrograms. They also used these assays to look at some protective effects of melatonin and showed that pretreatment with melatonin can mitigate pyrotypic cell death. They also evaluated ROS production in microglia and mitochondrial cells, along with recovery with membrane potential post-LPS. So to look at these results a little bit closer, you can see in Figure A the Western blot results. You can see that melatonin decreased NLRP3 inflammasome markers in the hippocampus, such as cytokine IL-1 beta. That's that heavy marker of NLRP3 we talked about earlier. It also lowered cleaved caspase-1 levels. The protein levels of NLRP3 markers and cleaved caspase-1 are shown in graphs B through E. Overall, this just shows that melatonin significantly suppressed NLRP3 inflammasome activity in the hippocampus of these LPS-treated mice. So moving on to some of this immunofluorescence staining with these beautiful pictures. Figure A here shows that the immunofluorescence staining of IBA1, a mitochondrial marker. It also shows that NLRP3 presence in the microglia. This third column here with the blue stain is just a nucleus marker, similar to DAPI. Graphs B through D show that in D here, you can look and see that NLRP3 is mostly expressed in microglia, and it was reduced in that melatonin-treated mice. And then in E, you can see here that the number of microglia cells was reduced in these melatonin-treated mice. And then in C, you can see that microglial intensity was decreased in the melatonin-treated mice as well. And in B, you can see that NLRP3 intensity was greatly reduced in those melatonin-treated mice, which correlates again to this gorgeous staining up above. So these are just more ELISA and Western blotting results that kind of go over what I just went over in these past two slides. Essentially, the takeaway from all this is that melatonin does reduce mRNA, protein levels, cytokines, and exposure and expression of both microglia and NLRP3 when treated with melatonin. So this is a little bit of that ROS data. So they used microglial cultures and they measured ROS in microglia and in mitochondria. So that pre-treatment, that 2 hours before LPS injection with melatonin, reduced production of intracellular ROS. You can see that in graph A. So that means that it reduced production of ROS from microglia themselves. And then mitochondrial ROS was reduced significantly with pretreatment for melatonin, which you can see in B here in this lovely staining. And you can also see in graph form in C. And then in membrane potential was restored when treated with melatonin. So you can see that in graph form here in D, and then again over here in E with that recovery of membrane potential. So moving on to the SIRT1 data, SIRT1 protein levels were measured by Western blotting and immunofluorescence, and then the co-effects of SIRT1, melatonin, and NLRP3 were measured in vitro. Quantitative real-time PCR, Western blotting, and culture were used to examine the pathways of SIRT1 and NRF2. So let's go over this just a little bit. So SIRT1 protein levels were increased. You can see that in the Western blotting in A, and then again in graph form over here in B. You can see that it's really specifically increased at 1 hour post-LPS injection. And in vivo, melatonin increased SIRT1 positive cells that were decreased by LPS. You can see that in staining over here in C, and you can also see it in graph form over here in D. Graphs E and F show that sirtuinol, which is something that completely inhibits SIRT1, reversed melatonin effects and increased NLRP3 mRNA levels, which you can see in graph G here. And then when SIRT1 was inhibited, that NLRP3 levels increase. So you can see that over here in graph H, and then in Western blot staining in I. Sirtuinol, which again just inhibits SIRT1, counters melatonin's protective effects on pyrotypic cell death, which is shown in graph form over here in J. So essentially the takeaway from this slide is just that SIRT1 goes up with melatonin, and when it's inhibited, NLRP3 goes up. So again, over here, this is just NRF2 and SIRT1, which were shown to co-modulate melatonin's effects on LPS-induced depression behavior. So this is that quantitative real-time PCR data, some Western blot data that was just showing that NRF2 is the pathway that SIRT1 is using to be able to co-modulate melatonin's effects. So in summary, melatonin is shown to attenuate LPS-induced depression-like behavior through the NRF2 SIRT1 pathway in the hippocampus, and melatonin downregulates NLRP3, inflammasome formation, activation, and activity in the hippocampus, as well as microglial inflammasome activation, ROS production, and pyrotypic cell death. Excuse me. So if you have any questions, please let me know, and then the source material is linked down below.